Warm greetings to all researchers, scientists, professors, and students. And on behalf of International Consortium of Water Researchers, I welcome you all. And the International Consortium of Water Researchers (ICWR) is a non-profit consortium established on June 5, 2020, on World Environmental Day, with an objective to connect water-based researchers and scientists working all over the world. It is promoted and supported by international and national advisory committee members of various research and educational institutions across the globe. And ICWR operates with an interest towards enriching knowledge among the global citizens at four different stages. And at water fellows, those who are having experience more than 20 years can join us as water fellows, and water experts, those who are having 10 to 20 years of experience, can join us as water experts, and those who are having less than 10 years of experience on water research can join us through young professionals in water research. And we are welcoming student members also to our network. And ICWR does this through organizing webinar series, training, and workshops on water-based modeling and annual virtual conferences. We also further our objective by operating effectively among the water-based research aspirants at all levels. And with this short introduction, I like to I would like to introduce Professor Eric from University of Paris, France, for our introductory webinar on anaerobic wastewater treatment and the importance of trace elements. And over here, uh, this is our first program organized from ICWR. And as an introductory part, Professor Eric is going to give introduction about uh, and deep insights about anaerobic wastewater treatment and the importance of trace elements. And he is having experience. He is from Paris Globe Physical Physics Institute, and he's having his research activities mainly focuses on study of metals and metalloids geochemistry in engineered ecosystems, especially in bioreactors, and mainly dedicated to wastewater treatment for pollution control and resource recovery, and investigation of the role of living organisms on the weathering of materials and minerals, use of biochar or hydrochar as additives in anaerobic digestion, and soil remediation or as sorbing materials for the removal of contaminants. And he's also working on advanced oxidation process in combination of soil washing with surfactants and addition of soil amendments such as biochar. He's having more than 290 plus research articles, 8 books, and 26 book chapters, and he's having a citation around more than 8,800 citations are there. And I feel very happy to introduce him to the community of water researchers. Professor Eric, now the platform is yours. You can share.
All right, thank you very much for this introduction, and I hope you are all hearing me well. And I'm quite pleased to inaugurate this series of webinars. I think I'm the first one to give a talk. So welcome all. So today I would like to introduce you to the field of anaerobic digestion and anaerobic wastewater treatment, and also present the importance of trace elements. So this is a field of research that I've been working on for almost 20 years now, and I'll try to give an overview of this field. All right. So the outline of my presentation, yeah, it's rather simple. First of all, the context of the presentation, why trace elements are important, and then I will speak about something that might be new for some of you, which is looking at trace element fate and origin in bioreactors, and looking at its distribution between liquid phase, solid phase, and understanding of trace element bioavailability. Of course, when trace elements are are added or come together with the feedstock, we have to understand what is the fate of trace elements, because those elements are not degraded in the digesters. And then I will conclude with some take-home messages. So the context. So we know that in digesters, we have three main phases, starting with the sludge, the liquid phase, and the headspace of the reactor. To feed this reactor, we are providing different type of feedstock depending on what is available and what it was designed for. So it may start with manure, crop residues, but we have many different types of organic feedstock, such as sewage sludge, household waste, and food waste, for instance. And all these organic compounds are going to be degraded. And while being degraded, many of these organic fraction will be converted to biogas, and then you will find the carbon as methane and carbon dioxide, and the rest will remain in the reactor as a leftover of the degradation process. For optimizing the process, very often we have to add micronutrients, which are meant to enhance the removal of H2S, which may sometimes be formed in anaerobic digester. Or we are also dosing trace elements to enhance the microbial activity. To remove H2S, sometimes people are also adding microdose of oxygen, which oxidize H2S into elemental sulfur and prevent the transfer of H2S into the biogas. So basically, in the end, after treatment, we have a digestate. So this digestate is a mixture of water and solid material. That's why we need to go through a solid liquid separation. And in the end, we have the liquid digestate that may be further processed for producing fertilizers. And solid digestate may also be further processed through composting, for instance. So if you are interested to know more about these aspects, I would recommend you to read this good review paper from Moller and Müller. So now, there is this strange things. Okay, I'm discovering Zoom as a webinar for webinar, but okay. So now I'm going to introduce the importance of trace elements. And first of all, we have to come back to this microbiology course, where you have been all learning that microbes need different types of nutrients that could be macronutrients, such as carbon, nitrogen, phosphorus, sulfur, so which are usually those high concentration. And also to ensure that the microbial activity will be well developing in the process, trace elements are also required, such as nickel, cobalt, manganese, iron. So there are many different nutrients that are there and which are very important to ensure there will be microbial growth and microbial activity. When we are referring to trace elements, we have two types of trace elements. Those which are recognized as essential and those which are recognized as not essential. So if you would dose these trace elements, so if you look at the trace elements concentration and its effect on microbial activity or microbial growth, you would face two different situations. So the one with essential elements is when the trace elements concentration is very low, so you would be in deficient conditions. And when the concentration is too low, the growth and microbial activity is going to be very low as well. So if you want to improve the situation, you will have to dose trace elements, and then in the end, you will reach a plateau, which is the optimal activity that you can obtain when you dose trace elements in your microbial system. But if you continue to dose this trace element in the reactor or in the batch system, in the end, what you get is that you can observe a decrease of the activity. So the effect is like toxicity effect, and then it becomes very much severe when the concentration is too high. So that's what we have for essential trace elements. But now if we move to non-essential trace elements, what you would see is that, first of all, for low concentration, there will be no effect. But then at some point, when the concentration is too high, then it gets toxic on the microbial activity, and then this microbial activity will decrease till reaching zero. So this is, for instance, the case if you would be using lead or cadmium. But here in that case, you would be like having like nickel, cobalt, copper, zinc. That would be that will be classified as essential trace elements. So now let's go to the origin and fate of trace elements. So as I just explained, trace elements are very important, but what about the situation in aerobic digestion? So we know that microbes require these trace elements, and it has been reported in many publications and even textbooks that nickel, cobalt, zinc, molybdenum, iron, selenium, and tungsten are very important for the archaeobacteria, which are involved in methanogenic activity. But we have also, it has been also reported, which is less known, for some potential deficiencies and importance of zinc, copper, and manganese for hydrolytic bacteria. So these are the two type of microorganisms that might be affected by the presence and the bioavailability of trace elements. I'm trying to remove that. I don't know if someone could remove. Let's see.
I think, Professor, there will be an option, annotate, I guess.
Yeah, I'm trying to click and yes. Okay. Now, because this was a bit annoying. Thank you.
Yeah, thank you.
Okay, let me continue. Okay, let's close it. Yeah, and yeah. Okay. So, so now let's have a look at the biochemical degradation of the organic feedstock, which may contain proteins, carbohydrates, lipids. And as you all know, this process is taking place in 4 different steps. The first step starts with the hydrolysis, where you can see that the bacteria involved in the hydrolysis and fermentation require zinc, copper, and manganese. So all these complex biopolymers will be converted to monomers and oligomers, which starts from sugar, amino acids, and long-chain fatty acids. And all these compounds will be used as substrate by the microbes involved in the acidogenesis. So all these compounds will be converted to short-chain fatty acids, but also at the same time, there will be a production of hydrogen and CO2 and production of acetate. These short-chain VFAs will be converted to acetate and as well as hydrogen and CO2. So this will be the step 3, where the acetogenesis will take place. You might have even conversion from hydrogen and CO2 to acetate, which are also involved. And from this step, so on which is step 4, the conversion of hydrogen and CO2, which is will be leading to methane and CO2, and the one from acetate. Sorry, this one is going to lead to the formation of methane, and this one, acetate, is going to lead to the formation of methane and CO2. So for these 2 types of methanogens, these trace elements are very important. So this is illustrated with this slide, with this, which is showing you where trace elements will be involved. As you know, the conversion of acetate or CO2 and hydrogen is not going to take place in a single step. There will be multiple steps where different enzyme will be involved, and we know that several enzymes would require the presence of trace elements to be active. And this scheme is very interesting because it shows you the importance of different trace elements. As you can see, we have a lot of iron, which is highly important to ensure that the enzymatic system is going to be active. But in some places, we can find that, for instance, cobalt is important here, nickel, and zinc is also important. And if we go here, molybdenum is also important. So this pathway is showing you for 3 different pathways all the trace elements could be involved in the production of methane and CO2. So this is a clear message showing that those trace elements should be somehow uptaken by microorganisms to ensure that they will be able to produce methane. So how does it affect real scale processes? So as you know, anaerobic digestion could take place either in a co-digestion system where you're going to mix different feedstock together. In that case, there are no much problems reported regarding the macronutrients concentration. So because you are mixing different feedstock, you ensure that you will bring all these trace elements. However, if you are running a monodigestion system, then the probability that trace elements limitation occur is very high. So the question is, where does these trace elements come from? And this is from IEA Bioenergy report, which shows you that depending on the type of feedstock, you can bring different concentration of trace elements. Of course, here you have essential trace elements like zinc, copper, nickel. However, you have also some toxic ones like lead and cadmium, as well as mercury. And you see that depending on the type of feedstock, the concentration of, for instance, nickel is very high when you're looking at a manure, but it is very low when you're looking at agri-food products, such as dairy waste, for instance, but also slaughterhouse waste streams could also contain very low concentration of nickel. So if you would run those systems only on one type of feedstock, you may have to face limitations, trace elements limitations. And this has been reported in a paper that was published in 2012 by Lindhofer and collaborators. So what they have been doing, they investigated 1,500 digesters in Germany and looking at the concentration of macro and micronutrients, and as well as looking at the VFA concentration. And out of these samples, they could see that 600 digesters were using energy crops only, and 80 digesters were using waste substrate. Okay. So in 60 plants, they could see that the micronutrients level was pretty low, and they decided to dose in the full-scale reactor trace elements to see if they would be able to find some changes in the biogas production rate and yield, and also looking at the VFA concentration. So this is what they did, and this is an example of what happened on one of the reactor. So this is the propionate and the acetate concentration in the reactor, and then around day 50, they decided to dose this trace element cocktail, which is here. So this is the trace elements they have been adding in the reactor. And then in a couple of days, they could see that the acetate and propionate concentration in the digester was going down, which is a good indication that all these VFA were probably very quickly converted to methane. And this is illustrated here through the second scheme, where you have the methane production yield, cubic meter per ton of fresh matter, which shows you that as soon as trace elements were dosed in the system, the biogas production yield increased very much. So this is a good illustration of what is happening. And at the same time, they could improve the plant capacity and reach somehow almost 100% of the plant capacity. So this is a good indication that at real scale, there are some issues with trace elements deficiency. So that's why in the literature, many people reported some concentration range for trace elements that they would be required to operate anaerobic digester in optimal conditions. And as you can see, for instance, I'm just taking one example here. The concentration for nickel, for instance, was ranging from, so the concentration are reported in milligram per liter, so which is 610 to minus 3 milligram per liter, but then you may increase up to 30 milligram per liter. So different authors reported different concentration range, but no one really agrees on what is the optimal concentration of trace elements to be added in a reactor. So why is that? Well, what you have to bear in mind is that when you dose trace elements in digesters, those trace elements are going to partition in different phases, in the liquid phase, in the solid phase, and there will be some kind of geochemical processes taking place there. And for instance, it has been reported that the precipitation of trace elements with hydrogen sulfide, which is very often present in digesters, is leading to precipitation of these trace elements. And at some point, it may reduce the percentage of trace elements added as being bioavailable, so being bio-uptaken by the microorganisms to be used in the enzymatic system which are involved in biogas production. So what is very important, if, as I said before, if you observe a trace element limitation in a biogas system or biogas process, it would be required, if you want to improve the performance of the system, to add a trace element solution. So now let's have a look of what's going on there. So this is an example of some work we have been doing a long time ago, where we looked at a USB reactor, and we have been running many reactor setup in different reactor conditions. And in the end, we were not able to link the total trace elements concentration or content of the reactor to the reactor performance. So we started to investigate what is the reason for that. And we know that the key player in USB reactors are contained in granules, where you can find these methanogens among other trophic groups which are important for anaerobic digestion. And then we started to investigate what is called the chemical speciation. So what is chemical speciation? This has been defined by UPAC, which is the International Union for Pure and Applied Chemistry. And this association has published some guidelines. So speciation or speciation analysis is reported to be linked to analytical chemistry, where you are aiming to identify and measure the concentration of one specific or one or several specific chemical species in a sample. And the chemical species are specific chemical forms of one element that could be from different isotopic composition, having different electronic or oxidation state, or having different complex or molecular structure. And this is very important, as I will show later, because this will impact the bioavailability of microbes, of the trace elements to the microbes. So this is where I start from now. So let's say you decide to spike trace elements in your bioreactor. So what is going to happen is that these trace elements is going to, in the liquid phase of the reactor, is going to partition between the inorganic chemical species that you have in a reactor, which is mostly sulfide, phosphate, and carbonate. And on the other hand, you have a high concentration of organic material, which have the capacity to form complexes with trace elements. So when you spike trace elements will distribute between these inorganic complexes and organic complexes. And in the end, what is remaining in is as a free trace elements might be rather low. So that's why you have to dose quite a significant concentration of trace elements in a reactor to ensure that you are going to maximize the methanogenic activity in bioreactors. So this is the boundary conditions that are already described in the previous slide. And then this is something that we did a long time ago, where we have been looking at the methanogenic activity of methanogens fed with methanol. And what we did, we looked at the methanogenic activity. So this is the BMP test with an online monitoring system. This monitoring system is showing you here the production of methane with and without cobalt. And of course, you can see that when cobalt is added in the system, there is a very important increase in the methane production rate compared to the system which did not receive cobalt. So somehow adding trace elements in the system will speed up the methane production rate in the reactor. So this is illustrated here with the methanogenic specific activity, and which shows that trace element limitation is affecting the microbial activity. So we decided to perform some tests. So what we did is that we collected these methanogens that were from granules. So we collected the biomass from a distillery cobalt deprived granular sludge. We fed this sludge with methanol. We have been controlling the chemical composition of the growth medium. And then we have been changing the cobalt concentration from 0 to 5 micromolar. And then to ensure that the cobalt concentration will be somehow kept stable over time, we use a complexing agent, which is EDTA. And then after that, we monitor methanol, we monitor VFA concentration, gas pressure, total dissolved cobalt, and pH in the system. So this is what we obtained. And so what we did is that we monitor the methane production rate, and then we convert it to the methanogenic activity, specific activity. And here is the table showing you the cobalt added. And as I forgot to explain before, we use this EDTA cobalt system to somehow calculate back what would be the free cobalt concentration in solution. So for this, we have been using a geochemical model. And then when you had, for instance, 5 micromolar of cobalt, then the free cobalt concentration is 1.2 10 to the minus 6 micromolar. Somehow it's 10 to the minus 12 mole per liter of free cobalt when you are dosing 5 micromolar of cobalt in your system. So you can see that there are several order of magnitude difference between the free cobalt and the total cobalt added in the system. So we have been looking at the results, and then we realized that at some point it was required to ensure a maximal methane production rate from methanol to have in solution 5 10 to the minus 13 mole per liter of free cobalt. so which is extremely low concentration and which is hardly measured by any analytical method that we have available on the market at the moment. Of course, now we may wonder till which concentration we can dose these trace elements. So if you are dosing trace elements and you are going beyond this concentration, what you are going to see is that you are going to see a toxic effect, which will be materialized by a decreased methanogenic activity. So concentration of trace elements which is too high leads to first a waste of resource and also toxicity on the methanogenic activity. So we know that in the past many people looked at toxic effect of what is called potentially toxic elements on anaerobic sludges, and then many people found out that and reported different concentration of total metal concentration. As I said, as the concentration were mostly reported on total metal concentration, they would never consider what was the metal speciation. So this is what we decided to do. So first we run the reactions. We run the BMP test as we did before on the previous case. And then we increased the concentration from 5 micromolar to 1,000 micromolar. And you can see obviously that for 1,000 micromolar, there is no methane production, but 5 micromolar is somehow kind of optimal when you compare to the other concentrations. So we wanted to know what is the free cobalt concentration, which is started to be toxic for the microorganisms. So this is what we started to look at the speciation of trace elements, and based on these, we started to measure decided to measure the free cobalt concentration in our system. So for this, we have been using what is called the Donan membrane technique, DMT. And this DMT is somehow a DMT cell, which you can see here. And then this DMT cell is somehow has two sides with a membrane in between, which is a cation exchange membrane. And then on the donor side, you are recirculating the BMP test system. And the other side, you have the acceptor solutions, where you should be able to measure or either directly or indirectly the free cobalt concentration, because only free cobalt can cross this membrane. So this is the setup with the different reactors. So these are the BMP, these are the DMT cells. We have here the peristaltic pump to ensure the recirculation of the different liquid from the donor and the acceptor side. And in the end, so what we get, this reaction, the sludge, the medium with different cobalt concentration, mixing, reacting, activity test, and then after some point we have been filtering, because it's very hard to operate the DMT system while the metallurgic activity is taking place. So we are been collecting only this after mixing and after activity. And then in the end, you implemented the DMT test. So we started to look at the methane production yield or rate as function of days. And then we converted this information as the percentage of inhibition. So meaning that we started from 5 micromolar, we estimated as optimal methane cobalt concentration. And while increasing the total cobalt in solution, you can see that the methane production start to be inhibited. So from this graph, we could measure the inhibitive concentration of 50% of the methanogenic activity, and this has been calculated as 90 micromole per liter. But this was calculated on the total cobalt added in the system. So we wanted to know more about the cobalt in solution. So the cobalt in solution is reported here as dissolved. And of course, when we have been reporting this, you can see that the concentration of cobalt is much lower than the one added in solution. So in the end, we are been ending up here, and we redraw the curve, and we could do the same after measuring the free cobalt concentration with the DMT device. So based on the on this value, we can see that the IC50 was here in that on that figure to be 22 micromole per liter. So we did that for different reaction matrices. So either adding cobalt with carbonate and phosphate, we calculated the IC50 on the total added. Then we did the same on the dissolved cobalt, and then we looked at the free cobalt concentration in solution. So what we ended up is that for 3 different systems is that the free cobalt concentration, which is somehow toxic for microorganisms, was somehow very close, and so we could get something which was on average about 13 micromole per liter, which was the free cobalt toxic for 50% of the metallogenic activity. So if you would start to compare then on different conditions, and you could see that when you consider the total cobalt added or the total dissolved cobalt, you have very high differences in the concentration. But when you look at the free cobalt, you somehow have a good evaluation of the toxicity based on the free cobalt concentration. So just to sum up, we indirectly measure for metallogenic activity from methanol. We could find out that free cobalt, which is optimal for the metallogenic activity, was 10 to the minus 13 mole per liter. So for these for 2.5 micromole added in solution. And then for the toxic concentration, it was determined as 13 10 to the minus 6 mole per liter. So which is responsible for 50% inhibition of the metallogenic activity. So this is an example to show you that there is a huge difference when you are looking at the optimal concentration and when you are looking at the toxicity for 50% for 50% inhibition of the metallogenic activity from methanol. Of course, there is still a lot of questions behind. Would we get the same results for other trace elements? We know that we looked for cobalt, but we can also look for nickel and other ones. What would happen if we change the substrate? And what would happen if sulfide is in your system? So that's why now we decided to move a little bit further, because as you could see from this graph, we could measure the impact of precipitation in solid phase. So solid phase is involved as well in the trace element partitioning through co-precipitation process, through adsorption and surface complexation. So these are different type of geochemical processes that will affect the partitioning of free trace elements in solution. So that's why we have published in 2016 a review paper, where we looked at the different methodological approach to understand speciation and bioavailability of trace elements in anaerobic digesters. So it's a paper where you have some good practice for measuring and determining the trace element speciation in digesters. And there we looked at different methods, and one of the method was looking at the trace elements and major element speciation in solid materials. So looking at biofilms, looking at sludges, and when you're interested to look at trace elements and so on, you may use electron microscope techniques. But if you are really willing to look at other more accurate speciation or chemical speciation information, you have to use spectroscopic techniques. So which have advantage and drawbacks, which I'm not going to comment now because we don't have much time. But yeah, so this is something that you can look back later on. I'm just going to remove this. Yes. Yeah. Okay, so this is an example of what you can do with spectroscopy adsorption technique. And this is an example of the speciation of sulfur. So you can get different spectra for different sulfur speciation in sludge sample, for instance. And this is an example when you start from the most reduced sulfur compound, which is sulfide, moving to the most oxidized sulfur compound, which is sulfate. So depending on the speciation of sulfur, you have a different spectral signature that you can, or fingerprint, that you can further use for knowing the speciation of these elements in sludge. One of the main drawbacks of this technique is that the detection limit for this technique is not going to low enough to analyze the speciation of trace elements in the solid material. So one option to approach the speciation or the fractionation is to start with what I will call the sequential extraction. So I'm not going to comment what is here, but the sequential extraction is somehow used to operationally define what is the distribution of trace elements in the sludge between the cell wall of the cell wall of the microbes and EPS, the carbonate fraction, the sulfide fraction, the phosphate fraction. However, it's not so simple, and the most advanced fractionation techniques that you can use is this one that we published already some time ago. Is that we, based on using different chemical, we can sequentially extract trace and major elements from the biomass, and then you can discriminate, or you can classify them as exchangeable, carbonate fraction, organic matter, and sulfide fraction, and residual. So somehow it gives you some feeling about the mobility, leachability of trace elements when they are sorbed on different solid fractions that are available in sludge or biofilms. So this is something that we implemented on, again, USB reactor. You can find all the results in this paper. So what we did was to look at the effect of the presence of sulfide in metallogenic reactor. So we compared two USB reactor running on these conditions. One reactor was running without sulfate addition, and the other reactor was running with sulfate addition, and this sulfate being, of course, reduced to sulfide in view of the low redox conditions of the system. Of course, we have been dosing all the trace elements needed for the metallurgens, but we forgot to dose trace elements in the reactor. So we dose cobalt at the startup of the reactor, but then we monitor the fate of cobalt in the system. So basically, what we got in the reactor without sulfide is that first we have a distribution of cobalt in different fraction: exchangeable, carbonate, organic matter, sulfide fraction, and residual fraction. And then as when the reactor was operated, we are been ending up to 130 days. So you can see that the concentration of cobalt in the sludge was going down very quickly. When sulfate was in solution, you could see that we could keep the cobalt concentration in the reactor for a longer period, and in the end, it was still slightly higher in the reactor than without sulfide concentration. So this is it was just used for comparison purposes to see how metals behave between two different systems. But from here, you can hardly get accurate information about the speciation. So you are looking at fractionation, so chemical fractionation of trace elements in sludge. So when you are able to use this sequential extraction to look somehow all the metals are distributed between these fractions, you know what is in solution, you may also be tempted to use geochemical models. So this has been developed by some colleagues from Linköping University in Sweden. And what they did, they have been running many reactors which were operated in sulfidic and ferruginous conditions. And then they looked at the trace elements and iron speciation in these reactors. So basically, what they started to do is that they looked at the speciation and of sulfur, because sulfur is known to be a very important element in immobilizing the trace elements. So they have been running this X-ray absorption spectroscopy, looking at different potential reference components that could be there, and by linear combination, they could find out that iron sulfide was the main sulfur species in the reactor. So that was a first aspect. They have been somehow looking at the sludge and seeing at the metal and iron fate in the reactor. However, it was not possible to do that for trace elements. So what they did is that they use a geochemical model, which is Visual Mintech. They have carefully selected the thermodynamic constant for metal sulfide, which is a bit tricky because they are not all reliable, but you can check their paper. They have been doing a great job there. And also in the model, they have been adding parameters that were easily to measure, like temperature, like pH, like the CO2 partial pressure for the carbonate concentration in solution, then the total concentration of iron, of trace elements, phosphate, chloride, and sodium. So they measure all these parameters and they input all these parameters in the model. And of course, they looked at the concentration, the total concentration of different elements, starting from cobalt, nickel, copper, zinc, but also cadmium and lead, which are also present in digesters, CSTR in that case. And then they also measure all these other elements. So I'm not going to go in details, but if you like to go back to this, you can check this paper. So they input all this information in the model. And then what they did, they looked at, for instance, the iron speciation in the reactor. So when they were operated these sulfidogenic bioreactors, they could see that iron was mostly present as iron sulfide. But then on the 3 other reactors that were more ferruginous reactors, they could see that iron was mostly present as siderite, which is iron carbonate precipitate. So by using this model, they could somehow find out what is the speciation of iron, and actually what you have here in the sulfidogenic reactor fits quite well with the previous results that they measure here with the scanning electron, with the X-ray absorption spectroscopy approach. So they also did that for trace elements. And what they could see is that in the sulfidogenic reactors, cobalt was mostly present as cobalt sulfide, as a mineral species, but they could also show the formation of cobalt thiols complexes, which are mostly related to organic ligand. When they were in ferruginous conditions, then you could see that phosphate and carbonates are taking are playing a major roles and much higher roles than in the previous cases. They did the same for nickel, and they could see that nickel in sulfidogenic reactor were mostly as sulfide precipitates, but also forming complexes with thiols groups from organic compounds. And for the other trace elements, they could find out that in the ferruginous system that phosphate and carbonate are playing a big role. So they looked at the concentration of free cobalt in their ferruginous system and sulfidogenic system. They looked at the same for nickel. And basically, if we look at cobalt, for instance, the concentration of free cobalt was somehow very close to the one that we measured in the methanogenic activity based on methanol as a substrate. So here you have something which is 10 to minus 10 to 10 to minus 11 mole per liter of free cobalt in the different reactors. In the sulfidogenic reactors, the concentration was was a bit the ferrogenic reactor was a bit different. But you can see that we have somehow quite similar results in terms of free ions in solution, which is considered to be the most bioavailable fraction. So they have been proposing a different model showing what would be the fate of iron, nickel, and cobalt in the sludge phase and also in the liquid phase of the reactors. And they could report that sulfide was playing a big role, and also the organic functional groups such as thiols are also playing a big role in the complexation, in the immobilization of those trace elements in solution. So I would strongly recommend you, if you're interested in this topic, to read this PhD thesis from Shakiri Yekta, which is very informative on the fate of nickel and cobalt in reactors. So, of course, we have something which is missing, because we have been looking at all of these here. But what we miss here is the markable activity. So all those trace elements in solution and the free fraction interacts with the methanogens. How can this be uptaken and somehow those trace elements being inserted in the enzymes for ensuring good biogas and good methanogenic activity? So one of the difficult point is to measure this free ion in solution. So we know that trace elements may enter into the cell through different routes, which is pretty difficult because you have to make sure there are specific transporters to ensure that the free metal will be entering the cell, and when it is in the cell, it will be used for some macro biomolecules and such as enzymes. But you may have also some conditions where microbes could synthesize complexing molecules that would help to uptake and acquire those trace elements and make sure they go back in the cells to ensure ensure their microbial activity. So this is still unknown. There are not so many research done on that aspect, but still something that people should dig in. And then if you are interested to look at the bioavailability of the system, you have to bear in mind that this is not a snapshot. This is not a snapshot. Everything is going to be highly dynamic system. So if one fraction of these free trace elements is going to be uptaken by the cell, all these equilibrium are going to be modified. So one of the tricky point here is to assess the bioavailability. So how quick the system will be able to provide to supply trace elements and bioavailable trace elements to the cell in the system. And this is something which is still under investigation in the field of anaerobic digestion. How dynamic is the system? How can the sludge and the complexes and solid material could supply the trace elements to the cell? And this is something which is very difficult. And there have been different attempts to assess the bioavailability. And recently, people started to work with what is called the diffusive gradient in thin film, which is a DGT. So this DGT is this capsule here, which has a membrane filter, which has a diffusive gel, which has a resin, which is able to accumulate. So to somehow mimic the interface between the cells and the liquid compartment. So by using this tool, people have been trying to somehow to understand how the medium could provide trace elements that could be easily uptaken. So to understand the dynamic behavior of chemical speciation in the reactor, people started to use such type of tool. I'm aware there are currently several PhD research done on that, but I did not see yet much publication, but probably they are currently preparing them. So I believe that will be something that will pop up in the next months or years, and you will be able to see people trying to use this DGT to assess the bioavailability. So that's somehow a kind of summary of the interaction of trace elements in reactors. People may also wonder, okay, if you dose trace elements in the reactor, what is the fate of these metals? So again, people try to do that by using sequential extraction, looking at water-soluble fraction, exchangeable carbonate, iron and manganese oxide, organic matter, and residuals. So this is a work done by this group who developed such sequential extraction procedure for anaerobic sludge. However, there is not a single information about the metal sulfide fraction, which has been shown in my previous slide to be extremely important. So these authors are not reporting the role of sulfide. However, you can see that already the organic fraction, which plays a big role here for copper, for instance, but we are only getting a rough estimation of the distribution of metals in the sludge. We are not getting real speciation or chemistry coordination chemistry information of this element in the sludge. And this has been done by some colleagues from France. What they did is that they looked at the effect of anaerobic digestion on copper and zinc speciation in sludge in different type of feedstock. And what they did, they looked at pig manure, they looked at the organic fraction of municipal solid waste, they looked at sewage sludge, and they even looked at sewage sludge mixed with green waste. And what happened, they looked at the metal concentration, so gram or milligram per kilogram dry weight before the digestion and after the digestion. So you can see that after digestion, wherever you look, the concentration of metals is much higher, which is of course easily explained by the fact that a significant percentage of the organic feedstock has been converted to biogas. So as metals are not going to be transformed, they are going to remain in the sludge. Their concentration in the digestate is going to be much higher than in the feedstock. Then they had a simple question: what is the effect of anaerobic digestion on the speciation of these two elements? So they started with zinc, and by using by using X-ray absorption spectroscopy, they could show that before, for instance, just taking one example here on sewage sludge, they could see that zinc was 53% sorbed on phosphate. Zinc was on for 22% zinc sorbed on iron oxides, and zinc was retrieved as zinc sulfide nanoparticles in the sewage sludge. So that's after the sewage sludge treatment process. But then all this sludge collected went to the anaerobic digestion, and you have a slightly change in the speciation here, showing that the percentage of zinc sulfide as nanoparticle increased to 30%. The share of zinc sorbed on iron oxide decreased to 13%, and the zinc sorbed on phosphate phases was increased a little bit. So this was not too big changes, but if you would looked at this, for instance, pig manure, you have a huge change where the zinc phosphate fraction is taking over. So this is some examples showing you how the anaerobic digestion is going to change the distribution of metals in the sludge. The same was done with copper, and here they could see, for instance, I'm just going to take this example, which is a mix of green waste and sewage sludge. So it was initially mostly organic matter, copper complex by organic matter, and copper as copper sulfide, which was 38%. But then when they performed the anaerobic digestion, in the digest state, the copper sulfide fraction was becoming slightly dominant over the copper complex by the organic fraction. So from this work, you can easily see that the sulfide fraction, the metal sulfide fraction, is taking a bigger role. And one might wonder, when you are taking this digestate on agricultural field, what would happen? Or the digestate oxidation, because you are taking the digestate from very anaerobic conditions to very oxidative conditions, what would happen to the metals? So this is a question, but in view of the time, I'm not going to be able to tell you the answer. But if you are interested, I have a couple of papers discussing that. Just some take-home messages for you, because I guess it's time to conclude. So first of all, we have trace elements in digesters, which comes together with the feedstock. But you can also add trace elements cocktails in digester to optimize biogas production yield and rate. So which might be a little bit interesting for the digester operating manager, if they are willing to treat much more feedstock and produce much more biogas. So that's is somehow kind of trade-off between the operation and financial conditions. We have also shown that trace elements total concentration does not correlate with toxicity or methanogenic activity. So toxicity and methanogenic activity are linked to the free metal concentration, so free concentration in solution. So that's something which is very important. However, when you are looking at the optimal operating conditions, the free trace elements ion concentration cannot be measured, because you are running you have concentration which is about 10 to the minus 12, 10 to the minus 13, which is often very, very difficult to reach by using a regular analytical method. So you have to use modeling for that. We also show that trace elements are distributed between liquid and solid fraction, that metals may accumulate in sulfide, phosphate, even carbonate, depending on the conditions. So depending on the feedstock, depending on the operating condition of the reactors, could be sulfidogenic reactors or somehow ferrogenic reactors. You have different situations. So that's something that you have also to bear in mind. And in the end, you could also see that trace elements concentration is always higher in the digest state compared to what you have in the feedstock, which is easily to understand because part of the organic material has been converted to biogas. That's the take-home messages I want to tell today. And for those who are really much interested in the topic, I've been involved with some colleagues, some European colleagues, in 2 books. A small one where we are giving some kind of decision tool or supporting tool, decision supporting tools for trace element dosing, trace element supplementation in anaerobic digesters. So trying to help operators to select the right conditions and decide when to dose trace elements in their reactors. And the other one is a book which is summarizing the activity that we had over 4 years networking program, which was dedicated to trace elements in anaerobic biotechnologies. And there we somehow wrap up a lot of information that you could find in this talk. And there are also many more information that you can also look at microbiology. There's a lot of things. These 2 documents are available from the IWA website. They are open access documents, so easily accessible to you. Well, please have a look. It's for free, and disseminate this document. And if you are really much interested in trace elements in anaerobic biotechnologies, I think this is the book that you have to read. So with this, I would like to conclude my talk, and I would like to thank you very much for your kind attention, and I'm ready to answer questions for a couple of minutes. Thank you very much.
Thank you, Eric. And Eric has shared his experience on anaerobic digestion and gave a lot of insights on the importance of trace elements in this digestion at anaerobic conditions. The comments from the attendees witnessed that. And uh, to have from Gugan Kumar, there is a question, sir. Sir, does trace elements concentration has any effect on persistent organic pollutant degradation? Are there any investigation on this?
Okay, that's a very interesting question. To be honest with you, I did not investigate this before. Of course, if the concentration of trace elements is too high, then you are generating toxic conditions, and then microbes involved in the degradation of the organic micropollutants are not going to be efficient. So that's one answer I can provide to you. However, I never came across any effect. It doesn't mean that it does not occur. But yeah, I think this has to be investigated.
Okay. And there is another question from our advisory board, Patnath Rahman from University of Portsmouth. And recently, coronavirus were reported in the biofilms of sewage. Will the loading for virus from sewage affect the anaerobic biomass in the bioreactor?
So if I understand the question is COVID, you want to understand?
Yeah, recently, coronavirus were reported in the biofilms of sewage. Will the loading of this virus will affect the anaerobic biomass in the bioreactor? It's like
Well,
cross contamination is
yeah, yeah. Well, I'm not a very big expert of viruses. I don't believe that this virus will interact with the microbial community which is there. This is not the right host for these microbes, but I believe there are other viruses that may play a role. But this is not, it's just a guess. I did not come across publication that would show any effect. So it's a good question and good research question. But I'm not able to answer this question.
And do you think is there any effect of co-addition to degrade lignocellulosic biomass in the process of anaerobic digestion?
There have been some research done in Sweden where they looked at copper, I think, and I can if the person who make this question can contact me further on.
Yeah.
I can communicate the work that has been done on that. So on the hydrolytic part, yes, we have recognized that some enzymes require cofactors such as copper to operate well. So indeed, there is a need for that. I have to say that there have been much more attention to the metallogenic steps and not very much to the hydrolytic step. And I believe there are still some work to be done here. Not sure how this will fully improve the lignocellulosic degradation in the reactors. I don't believe that's real key, but then it contributes to it. So we know that the hydrolysis is the limiting step in anaerobic digestion. So yeah, but it's more about bioavailability of the substrate for hydrolysis, which is playing a role.
Thank you, Eric, for your time. And there are some more questions are there. We will send you to your mail. So in
Okay.
your free
Yeah.
time, you can answer, and then we
Yeah.
will forward
Okay.
back to the attendees.
Great. Okay. So thank you very much
Thank you so
for
much for your.
organizing this event. I hope yeah, everything went well. I could maybe I could get easily understood by the people from the audience. And yeah, if you have questions, don't hesitate to contact me. We'll be happy to answer. And hopefully we can meet in real one of the next months when I will be traveling again to India or other places.
Yeah. Thank you so much,
Thank
Eric.
you.
Okay,
Have a
thank you,
nice day,
Yeah,
and see you very soon.
yeah. See you. Thank you.
Goodbye. Bye
Goodbye.
bye.
Bye bye.